Okay, good morning, everyone. First of all, thank you for giving me this wonderful opportunity. I am Carla Cervini, Academic Fellow in Food Mycology at FunShield University, and I will present today on molecular biology techniques for fungal identification with a focus on Aspergillus. So this is the outline of my presentation. I will start introducing the Aspergillus genus. Then I will explain shortly what are the traditional cultural methods, and then we're going through the polyphasic approach for identification, with in particular a focus on the molecular identification of fungal species, starting with DNA extraction, PCR amplification, DNA barcoding, and finally the conclusions. Although I guess you might be already familiar with the Aspergillus genus, I think it's still good to start with a short introduction. So they are known, these fungi are known for their economical and medical relevance. They've been described for the first time in 1729 by Micheli, and they are so called because of the structure which resemble an aspergillum, that is a Catholic tool used in the church. And they are known for being the most abundant and widely distributed organism on Earth. And they have positive effect because they are exploited by industry for the production of medicines and also for producing enzymes and acids, but they are mainly known for their negative impact. In fact, they are responsible of food and feed spoilage, and they are human and animal pathogens, and we are here talking about them because of their food safety impact, as they are known to produce toxic compounds which are called mycotoxins. So in this slide, I try to summarize the main mycotoxins or secondary metabolites produced by the Aspergillus species. The mostly known mycotoxins are the aflatoxins. These are B1, B2, G1, G2, and M1. And then ochratoxin A, patulin, and many more. However, among all of these, and there are also others that I haven't reported, only the aflatoxins, the ochratoxin A, patulin, and citrulline are regulated by the Commission regulation, the European regulation of 2023/915. All the others have no regulation because their effect on human and animal health is still under investigation. So here is a timeline of the Aspergillus taxonomy. So first studies regarding the characterization of the Aspergillus species are dated back to the 1975 with a paper by Rapper and Fennell, where this genus was where species of this genus were gathered in 18 groups. And since this moment till now, let's say, there has been different studies with lots of updates about the Aspergillus genus with, let's say, we started with 18 groups, and this has evolved till now, where with a recent paper, we have now a total of 6 subgenera and 28 sections and 76 series and 453 species among the Aspergillus genus. And over the time, there has been, let's say, a critical moment that is the introduction of the PCR methods that happened around the mid of 80s. And before these, before its introduction, let's say the identification of the species was mainly based on cultural techniques only, while since PCR has been introduced, there has been many studies which have led to what I just said now. So it led to the identification of 453 species. So we have now more possibilities of accurate identification of species by using a molecular approach compared to what was possible to do based only on traditional cultural methods. So, but still, it's still important to know how this traditional approach is conducted. So these traditional cultural methods are relying on the identification of morphological features of fungi, basically combining macroscopic and microscopic characteristics of these species. So here, first of all, is an overview on the most important macroscopic features of the Aspergillus species. So let's say that these methods are based on the use of different culture media and different incubation temperatures that will lead to the possibility of identifying the species based on the colony color and the texture of the fungal colonies, as reported on the picture on the left side, but also on the colony diameters. So we can measure the fungal growth of the species, usually after 7 days, and this may vary, and this varies depending on the media and the temperature. So the combination of different media and different temperature is giving us the possibility of discriminating among the species. And then mycelium column, the presence of exudate, and also the reverse of the reverse color of the colony can be checked as well, together with the presence of soluble pigments and also sclerotia. Sclerotia could be a very important feature, especially for some species like the Aspergillus section flavi, where the Aspergillus flavus, that is the most representative and known species among this section, can be distinguished among large sclerotia producer and small sclerotia producer. So is also another important criteria. In combination of these microscopic characterization, there is also the microscopic one. So this is usually conducted on slides on lactic acid, 70%, and the use of a microscope. And then we're going to see some specific characteristic, for instance, the seriation of the conidiophores, together with the conidial shape and the size, to be able, for instance, for the Aspergillus, to distinguish among uniseriate and biseriate fungi. And also other parameters that can be checked includes the shape of the vesicle, then the stipe or the presence of the Ewell cells, the cleistothecial wall or ascospores. And the combination, as I was saying, of microscopic and microscopic features will give us an idea of the species we are talking about. However, to conduct a morphological characterization which relies on macroscopic and microscopic features is not an easy task. So it requires a lot of expertise and lots of knowledge. It's time consuming, and sometimes some species are so similar among each other that this may lead to a misidentification or failure of a correct identification in most of the cases because they are so similar that we can't really be sure that we have identified correctly a species. So this is a traditional approach, and in combination to morphological approach, we can also use the extrolight analysis. So in this case, we're looking at the production of the secondary metabolites by the species. So this also can be done by using qualitative methods and quantitative ones. So qualitative method for the Aspergillus, for instance, relies on the detection of a blue fluorescent halo on a coconut agar medium. So basically, species of Aspergillus that produce aflatoxins will produce a fluorescent halo that will be visible at 365 nanometer wavelength on the reverse side of this type of media. Another type of, let's say, qualitative method that can be used to identify Aspergillus flavus and Aspergillus parasiticus species is the use of the Aspergillus flavus and parasiticus agar medium. And on the reverse of this colony, we can see orange halo if the species are producing aspergillic acid, and otherwise, if species are not flavus or parasiticus, we won't see this bright orange halo. And this is shown here in the central picture. So on the left side, we have the bright orange halo, and on the right side, we have a brown halo, which indicates respectively the Aspergillus, a potential Aspergillus flavus or parasiticus species, and on the other side was an Aspergillus tamari, so a non-aspergillic acid producer. But nowadays, the most used technique for the identification of the analysis of their secondary metabolites produced by fungi is with the LC-MS, and to quantify really the amount of mycotoxin produced, we include some standards, and then this way we can really have a multi-mycotoxin analysis that is currently the most reliable methods for mycotoxin detection and quantification. In this slide, I have reported an example of the different chemotypes. So for chemotypes, we intend the classification of the species, and this is the specific case of the Aspergillus section flavi that have been classified based on the production of these different secondary metabolites. So usually, to be able to distinguish between, let's say, by using chemotypes and morphological feature is already, could be already good enough to give us an idea of the species we are talking about. However, there might be some species that, on the top of being similar for their morphological feature, can also have some similar secondary metabolite patterns that in the end we may not be able to distinguish among them. For instance, if we look at these panel, this can be the case of some species such as the Arachidicola or Luteovirescens that are producing the same patterns of mycotoxins, and on the top they may be similar, and then at the end we may lose a little bit of confidence in what we have in front of us. So, and in this regard, for providing a highly confident approach to identify the species, Samson and et al. In 2014 have proposed the polyphasic approach for Aspergillus identification, which basically includes the morphological characterization that I just spoke about previously, together with the extrolytes. So these are still there and still very important for the identification, but might not be sufficient for a highly, let's say, specific and with a high level of confidence identification. Then Samson have proposed the molecular also identification. So a polyphasic approach basically consists of morphological characterization, secondary metabolites identification, and also molecular identification. So the molecular approach for fungal identification consists of, first of all, we need to isolate the fungi. So fungal isolation is a process very specific that can consist basically on the use of specific type of culture media. So I'm not going to spend a lot of time on this because it's more related to another technique. So I will go directly to more information on the molecular approach. But once fungal isolation has been conducted, we have to purify the fungal colony. So purification, to be sure we don't have contamination, is also a critical moment in the for the further stages of the identification. Otherwise, if the colony is contaminated, this may lead to some misidentification. So we have to purify, and purification can be also conducted with different methods. Usually, we can subculture the colony till we see that the colony is clean and we don't have any other fungal growing close to the colony of our interest, or through, for instance, a single spore purification approach. Once we have our colony and we are sure that it's pure, we can store it in glycerol stock, for instance, and then maybe when we're ready, we can continue with the DNA extraction. So we subculture the strain, we extract the DNA through an approach that we'll see later. And then we continue with PCR amplification and gel electrophoresis. Then we're going through sequencing and finally through bioinformatic analysis, which will include sequence editing and species identification. So now we're going through mainly the stages of the molecular approach more in details. So here we have DNA extraction. So the DNA extraction is the most critical point for accurate identification application. And this is important because fungal cell wall is complex and is composed of chitin, glucans, lipid, and is extremely resistant. So the overall goal of the DNA extraction is basically to be sure that we destroy very well the fungal wall, and all the DNA extraction protocols are based on these. So are based on the optimum fungal wall lysis that can be done through physical, chemical, or enzymatic approach. In this table, there is like a summary of the most used protocol for DNA extraction based on published paper. And some of them relies on the use of a commercially available kit for DNA extraction. Others relies on the use of the hexadecyl 3-methylammonium bromide. So that can be a laboratory-based method where we have to prepare some solutions, but it's also reported to be very efficient. And we've been used this method at Cranfield University a lot. And overall, so the stages of DNA extraction, regardless if we are using commercial kits or laboratory-based methods, are usually, they consist of the sample preparation, then cell lysis that can be done through the use of the CTAP, that is a detergent, or through mortal and pestle in liquid nitrogen. So with the goal at this stage of destroying the wall of the fungi. Then we have the protein removal stage, which it can be done through the use of chloroform and isoamyl alcohol mixture. Then DNA precipitation, that usually consists of the use of isopropanol. Then we have different stages of rehydratation with the DNA binding, and finally the washing stage that are usually done with ethanol, and finally the DNA elution that can be done through elution solvent, or sometimes also water can be used. Once we have our DNA extract, we have to check we have to check the quality of our DNA, and this can be done by the use of a spectrophotometer like the nanodrop, that is the one reported here in the picture. And the quality check is done through reading the value of absorbance at two ratios, 260 and 280, and 260 and 230. So this ratio had to be into precise range of values. So 260 to 180 values needs to be within 1.8 and 2. 260 to 130, the ratio needs to be between 2 and 2.2. And if the values are within this range, then we can say that our DNA extraction has been good and our DNA extract is of good quality. And then we can also take note of the concentration of our DNA. After this, however, to be really sure that we have a good DNA extract, we can also double check the presence of bonds by going through agar electrophoresis gel. So the agar electrophoresis gel will show us the DNA bonds that will move based on the principle of that small fragments will move faster than large ones, and also our DNA fragments will migrate from negative to the positive side of the gel. After this, once we have reached the stage of being happy with our DNA extracts, we can conduct the PCR. So the PCR analysis is mainly composed of our DNA sample, then primers, so forward and reverse primers, then nucleotides, deoxynucleotides, and Taq polymerase, so the enzyme, and finally a buffer. So here is a picture of a common thermocycler where the analysis is run. And here on the right side, we have an example of just some of the most common stages is an example of the most common, let's say, cycles of a PCR analysis that mainly consist of an initial denaturation stage and then a cycle of denaturation, annealing, and the extensions that needs to be repeated several times. So here it can be from 30 to 40 cycles, depending on the specificity of the primers, for instance. And here is an example of the principle of the PCR analysis and how it works. So it's based on the exponential amplification of a fragment of our interest and is used to make millions to billions of copies of a specific DNA samples very rapidly, and to amplify a very small sample of DNA to enable further analysis and molecular analysis. Very important component of the DNA of the PCR analysis is the DNA polymerase. So the DNA polymerase is the enzyme that will be able to incorporate the nucleotides to extend primers at their 3' ends in the 5' to 3' direction. In 1970, there was the Klenov fragment that was used commonly to perform PCR analysis. However, the use of this fragment of DNA polymerase 1 from E. coli was associated with some disadvantages. It's sensitive and was easily destroyed at high denaturation temperature. And it was late in the 70s that the Taq DNA polymerase has been introduced and is still now used and is known for being thermostable, higher sensitivity and specificity, and higher yield. And it's not a case that has been like declared as the molecule of the year by Science in 1989. Once we have done the PCR analysis, we can move towards verification of our PCR products on agarose gel. So what we can do is we can load a certain volume of our PCR products into an agarose gel and let it run. So it's good practice when we're doing this to include a positive control. So let's say a sample that we've tested and that we know for sure that is able to amplify for that specific couple of primers that we used, together with also a negative control. So a sample that doesn't have any DNA sample, so it can be just water. And then a ladder. So the ladder will give us the idea that our PCR products are specific for the sites that we are targeting in our PCR analysis. And more precisely, for Aspergillus species, the commonly used primers for the identification of Aspergillus species are the ITS, so the internal transcribed spacer, that is considered the DNA barcoding of Aspergillus, is the official DNA barcode in general for fungi. It has been considered and classified like this in 2012. And it's always a good practice to include ITS sequences when new species are described. And it's basically this region of the nuclear ribosomal, which include the ITS1 and the ITS2 regions, which are separated by the 5.8 small subunit situated between the 18 and the 28 subunit of the nuclear ribosomal DNA. And it's considered the barcode, so it's always good practice to include it when new species are described and when we are conducting fungal identification. However, sometimes it does not contain enough variation for distinguishing among species, because this region is present with a high number of copies per cell, and it's a highly conservative region. So we can be sure that we are talking about fungi. So it's giving us an idea that we're talking about fungi, but sometimes it might not give us information to be able to distinguish among species, as I just said. Then other possible markers have been considered for fungal identification, and in particular for Aspergillus identification. And these are the calmodulin. Calmodulin is considered the second marker for Aspergillus identification because it's easy to amplify, and there is also a database almost complete for all accepted species. Then there are also other markers that can be possibly used for Aspergillus identification, and these are the RNA polymerase second largest subunit. However, this is not easy to amplify, and sometimes it ends up with not having any good amplified product, or the beta-tubulin that is easy to amplify and that has been reported. However, it has been reported to vary in the number of introns, and PCR results in the amplification of paralogous genes. So here is a summary of the primers and PCR conditions for Aspergillus identification that you can have for you, and this is taken from Samson paper, the same paper that introduced the polyphasic approach for identification of the Aspergillus. And then once we have reached to this stage and we are happy with our PCR product, I'm still not sure that I know which species I'm talking about. So let's say that now we have to pass from the gel products, so from the PCR amplification, towards having this. So that is like a sequence of nucleotides. How do we get then from the DNA, from the gel to the sequence? So this is done through sequencing. So usually the Sanger sequencing, that is also called chain termination sequencing. So this method is used for, let's say, for reading the sequence that we have. It consists of 3 stages. So first of all, we are running a PCR with fluorescent and chain terminating deoxyribonucleotide. So this will be introduced randomly together with a mixture of deoxynucleotides as in a normal PCR analysis. Then we have the second stage, which is the sites separation by capillary gel electrophoresis, where basically where the fragment will be separated. In automated Sanger, all oligonucleotides are run in a single capillary gel electrophoresis within the sequencing machine. And then finally, we have the laser excitation and detection by the sequencing machine, where we're going to have our chromatograms with the different sequencing that we're going to be able then to analyze to understand what we have. So this will take us to this at the end. So we will have our sequencing and we will have our chromatograms with all these peaks. And what we have to do next is to basically read our sequencing. Our sequences. So first of all, before reading them, we have to spend a certain amount of time to clean and to edit our sequences. So we need, first of all, to have a software to basically conduct this. I usually use BioEdit, that is free and of easy use, and also there are lots of videos on YouTube that can help and support conducting these analyses. But just to summarize the main stages that we have to go through to have at the end a good sequence, these are basically trimming the reverse and forward sequences at the beginning and at the end. So it's usually at this part of the sequencing, so at the beginning and at the end of the sequence, that we have some unspecific results. So that we have first of all to remove and clean. Then we have to create a reverse complement. And finally, we have to assemble the sequences. Once we have done this, and this is very, very, very summarized, but let's say it takes a while to conduct this analysis. Once we have the clean sequence that has been through trimmed, we have the reverse complement done, and we have assembled the forward and reverse fragments, we can assemble, we can copy and paste the clean sequences into a Word file or a Notepad document and save it. And then once we have it, we can go to the BLAST analysis. So BLAST is the basic local alignment search tool of NCBI. And basically, once we enter our sequence into this database, we will have our results. So we will have our sequence that we have gone through BLAST, let's say, through matching with the available sequences into this database that will tell us basically what our sequence is, which species is. So through basically having clear what these different sections of the BLAST are. So here I highlighted some of the most important, let's say, parameters that is important to know how to read before to be sure that we know what we are talking about, and to be sure that we are conducting a good analysis. And for instance, here is highlighted the query cover, that is the percentage of the query sequence that overlaps the reference sequence. And to be sure that we have a good result, this should be higher than 70%. Of course, 100% query cover is desirable, but 98, 90, 99 are also good. Then the percentage of identification, this is telling us the sequence similarity, and usually this should be around 98 and 100% if we have a good sequence, and to trust our results with what the BLAST analysis is telling us. And basically, these results, the results of our sequences are usually compared to species that are available into this database. And also you can find the accession number. So this is a unique identifier assigned to a record in the sequence database. And together with this, the accession length. So it's telling us basically what's the length of the sequence of the species that has been reported with that accession number on that column. However, the issue of using BLAST is that sometimes there may be some sequences that have been deposited into this database but have not been verified, and so this can lead to some issues in the identification. And over the year, there has been, let's say, some progresses in making sure that we can trust our result. So, and one solution is, for instance, the RefSec. So this can be selected into the NCBI database. So as we're doing the BLAST, we can select the RefSec that will give us just ITS sequences that have been, let's say, evaluated and that we can trust. Or also the Mycobank database is also a reliable database, much more reliable than the BLAST, because all the sequences that are here present have been verified and examined by scientists, and therefore we can trust more the results by using these other database. Or usually a combination of both can also give us a good idea of our results, if we can trust it or not. So here, coming to the conclusion, so the identification of the Aspergillus species is very important to understand the risk of exposure to mycotoxins compound in food. Some species, such as those within the Aspergillus section flavi, are very similar based only on morphological features. And that's why polyphasic approach, including molecular characterization, is recommended to perform a right fungal identification. And within this, when conducting a molecular identification, we have to use trusted databases of sequences that have to include reliable sequences to be sure that we can trust our results. And with this, I reached to the conclusion of my presentation. So this presentation, of course, is part of the FunShield4Med European project, for which Cranfield University has received funding from the UK RAI and Horizon Europe funding program. And I'd like to thank you all for the attention. And if you have questions, please don't hesitate to contact me, to contact me. And thank you all, and see you soon. Bye.